but because of the COVID regulations, we decided to postpone my visit until the summer. I'd like to talk about work we're doing on wheat diversity. I should say this is not my main research interest. My main research interest is actually diet and health. We're particularly interested in improving the quality of particularly white flour for diet and health. But over the years, we've also become interested in diversity, particularly in relation to comparing the compositions of different types of wheat. And so my title is Comparing the Composition of Modern and Ancient Wheats. I need to start with some background about wheat. And we're interested in these green boxes on the right-hand side here, the cultivated wheats. About 90% of what we grow, or 95% of what we grow globally, is bred wheat, fitting east of them, hexaploid bred wheat. Of the other 5%, almost all of it is pasture wheat, so sort of tetraploid pasture wheat, AA, BB genomes. These are advanced sort of modern wheats. They've been subjected to a lot of plant breeding, and they're very successful cultivars. We also grow small amounts of other types of wheat, what are generally called ancient wheats. There's actually 3 types which are reasonably widely grown: einkorn, the original progenitor of the A genome of bred wheat; a form of tetraploid wheat called emmer, which is related to pasture wheat. It's a different subspecies of the same species; and spelt, which is the most widely grown, and that's a subspecies which is actually related to bred wheat, a subspecies of the same species. You can see the ancestry here. I don't want to talk about the way they're developed, but effectively they're developed by hybridization of starting with a diploid with different types of wild grass to produce tetraploid and then hexaploid. Having said that, we think probably that einkorn and emmer are actually sort of directly descended from types of wheat which were grown perhaps sort of many thousand years ago. Spelt itself might be a more recent development, and not so closely related to some of the ancient types. So just to show you what the heads look like, this is modern bred wheat, modern durum wheat, spelt, the ancient form of bred wheat, the same species as I said, and these ancient forms of the A genome diploid einkorn and emmer, the A genome tetraploid. Heads aren't shown to scale, and in fact the bred wheat head tends to be a lot larger than some of the others. At the moment we grow about 750 million tonnes of wheat a year. So that's the third amongst the cereals in terms of production after maize and rice, but it's the most widely grown and consumed cereal in the world. This is a plot from Wikipedia showing the current distribution of wheat cultivation. You see it's grown everywhere from South America right down in the south here, through to Scandinavia, Australia, and in upland regions in tropical countries. As I said, of the 750 million tonnes, so 95% bred wheat, about 5% durum wheat. But it's actually spread over this whole area over the last sort of 10,000 years. We know a lot about wheat, certainly bred wheat, because it's the only cultivated cereal which has never actually grown in the wild as a hexaploid species. It actually developed in cultivation, probably in southeast Turkey, about 10,000 years ago. And I put approximate dates here for the way it spread from southeast Turkey through Asia, reaching Japan by about 3,500 years ago, through North Africa, reaching Spain and Portugal about 5,000 or 6,000 years ago, and reaching the UK about 5,000 years ago. We know the introduction to Mexico, 1529. We know it's introduced to Australia in 1788. So it's actually migrated over the whole world in the last 10,000 years. And this migration is associated with adaptation to local conditions. Wheat is very adaptable, so possibly because it's a hexaploid and therefore has more plasticity in the genome, more flexibility. And as it's spread and adapted, it's developed massive diversity. There's probably at least 50,000 genotypes of bred wheat available. These have all developed over the past 10,000 years. So it's extremely adaptable, extremely sort of plastic genome, and now extremely diverse. So we have a lot of diversity to play with if we are wheat breeders. It's been bred scientifically for coming on for 100 years, but of course it's probably been bred for 10,000 years. because it's undoubted that the early farmers would actually have kept some of their best seed and they would have planted them again in the following year. So they weren't doing scientific breeding, but they were actually starting to preserve the best material and to continue to grow it. Scientific terms, the end of the 19th century, we started actually selecting from traditional types of wheat, which we call land races. Land races are generally not pure cultivars, they're sort of mixtures of seeds, but they're developed as adapted to different environments. So they've started working with land races and started to pick out the best types of wheat using empirical breeding. In the early or the first half of the 20th century, more scientific breeding. But then really after about 1950, the second half of the 20th century, we started using real scientific breeding, improved agronomy, and of course from about the 1990s, particularly the last 20 years, using molecular breeding to improve the efficiency and the speed of the process. This has been associated with increases in yields. This is yields in the UK, tonnes per hectare, only up to about 2,000. But in fact, our yields at the moment still averaging about 8 tonnes per hectare. There's actually various reasons for this, which I won't go into today. But you see over 100 years, over the first 100 years, increase from about 2 tonnes per hectare to 8 tonnes per hectare. You can see this in one of our experiments at Rothamsted. At Rothamsted, we've been carrying out field trials since the 1830s. We've got over 175 years of field trials, and we've grown cultivars on the same land with different fertilizer treatments. And you can see these different cultivars along the bottom here, which have been grown with changed at regular intervals to use the more modern cultivars. You can see that the yields were pretty well stable at about 2 tonnes per hectare until the 1960s and 70s. And then we had the introduction of different agronomic practices, but also the application of plant breeding, scientific breeding, and particularly from the about 1970 onwards, the introduction of shorter cultivars. This was the green revolution, the introduction of the semi-dwarf wheats. And from then onwards, we got a take-off of the yields from about 2 tonnes per hectare to 6 tonnes per hectare. And then nowadays we go up to 8 to 10 tonnes, depending on the year. Interestingly, this blue line here are plots which have received no fertilizer for 175 years. We still take off about 1 tonne per hectare, half a tonne to a tonne per hectare. And in this case, the nitrogen probably comes from mineralization from the soil and also from atmospheric deposition. So there's enough nitrogen to support about half a tonne to 1 tonne per hectare. The average yield globally is about 3.5 tonnes. So the global yield is about here. But in the UK, we go up to 10 or 12 tonnes in some years. So plant breeding has been very successful in improving wheat. We've improved the yields. We use breeding for genetic improvement, combine this with agronomy. And we've selected modern yields for 2 things: for high starch content. 80% of the grain is starch, which means that if you have larger grain, that's mainly due to the increase in starch content. And improved processing quality. Because most of the wheat consumed by humans is used for bread making, we're interested particularly in increasing the gluten strength. So modern wheats have been bred for yield and for processing quality. And it's been suggested that this has had detrimental effects on various things. Firstly, we use a fairly narrow genetic base. So the genetic diversity is decreased, but also probably more concern to consumers is to suggest that these modern wheats have lower contents of important components that we get from wheat in our diet, particularly vitamins and minerals, also bioactive components, also higher contents of gluten, which may be the case because of the gluten strength, and resulted to increases in tolerances, allergies, sensitivity. Also suggested that the modern wheats, because they have higher starch, have a higher glycemic index, therefore higher digestibility, and therefore contribute to increase in type 2 obesity, sort of type 2 diabetes and obesity. I'm not going to talk about these health aspects today. We do have interest in the area, but I'm going to talk more just about the broad diversity and whether the diversity has changed. It's important to know this because wheat, and particularly bread, is an important part of the diet. In the UK, we have quite a balanced diet. You know, we eat a lot of vegetables, fruits, also meat, etc. as well as wheat, but we still get 10% of our energy from wheat, from bread, and we get 20% of our dietary fibre from bread, about 40% from all cereals. So it's important to know whether some of these components are going down. Folate, vitamin B9, we get about 10% of our folate from wheat, from bread, also other B vitamins, and 10 to 15% of our mineral micronutrients. So if these components are going down in bread and in wheat, we need to know about it. So we've tackled the problem using different approaches. And we use different approaches partly because it's actually very difficult to obtain research funding for this type of research. So we've put together data from different studies that we've been involved in. We can look back at archive samples. I'll show you the Bull Book continuous wheat experiment. We have samples I'll show you in a minute. We looked at global diversity screens, including one in an EU project known as Healthgrain, which we participated in. We compare UK adapted wheats grown in field trials, and we compare modern wheats with land races. This is to compare older and modern types of bread wheat. Also, we made some comparisons of ancient wheat and modern wheats, again from Healthgrain, and then a field trial we carried out as part of an ongoing project I'll talk about at the end. So what I'll do is go through data from various sources and try and give you an idea of to what extent modern bread wheat actually differs from older types of wheat, and whether modern wheats are different to ancient wheats. So at Rothamsted, we started growing wheat in 1843, and we've preserved samples from all the field trials since that time. So we have archives. We have an archive of 300,000 samples of grain, soil and straw. We have duplicate collections, so we can actually go back over the material in our archives. This is quite a good resource for various types of work, but it has its limitations because all the material has been oven dried. So it's very good for looking at minerals. You can see the content of lead in wheat here has gone down as we've reduced the lead in petrol. Things like dioxins and furans have increased. We can even see nuclear accidents by looking at radionuclides in the wheat collection. So we can go back and analyse it, and it's most useful for things like minerals. But this is some data published over 10 years ago from some of my colleagues at Rothamsted, where they've actually looked at the mineral content of wheat from the archive. Now, this shows the yields of those plots from 1840 to 2000. As you can see, the yield of these high fertiliser plots has increased up to about 8 tonnes per hectare. What happened at the same time is the harvest index has increased, so we have bigger grain compared to straw. But we do see decreases in zinc and in iron, and other people reported this, and we get decreases really from the introduction of the semi-dwarf wheats. So the shorter types of wheat, which were introduced from the 1970s and onwards. We used to think this was due to yield dilution. I mean, if you have more starch, it actually dilutes everything else in the grain. So you dilute minerals by adding more starch to the grain. This is not the case, because when we look at some of our plots which have no fertiliser, that's the control here with no fertiliser, we still get a decrease in iron and zinc. So it's not yield dilution. Yield dilution may contribute, but it's not the sole factor. We think in this case the wheats which are shorter may actually have smaller root systems, and they're probably less good at removing iron and zinc from the soil, and therefore we get lower amounts of iron and zinc in the grain. And there's been a lot of work on minerals in wheat, and this decrease of semi-dwarf varieties has been recognised by other people. Having said that, we think it's probably the only reproducible change that we have seen in over this time period in the wheats that we grow. First one we looked at this was part of an EU project called Healthgrain, which was 2005 to 2010. As part of it, we grew 148 red wheat lines. We grew them on one site, just one site for 1 year. So single plots, no replicates. I'll talk about that later. In fact, most were from Western Europe, but they weren't all from Western Europe. You can see the distribution here, and the numbers relate to the size of the spots. So we had them from Australia down to South America, but a lot from Western Europe. And most of the lines were from 1960 to about 2004. But it did give us an indication of global diversity. We carried out a wide range of analyses by a European consortium. Our work, we're interested ourselves in dietary fibre, but also work on phenolics, particularly phenolic acids, alcohol saucinols, which are phenolic lipids. Work on terpenoids, including tocols and sterols. Tocols include vitamin E, and we also analysed for folic acid vitamins, so vitamin B9. What we saw was very little relationship between the content of these components and the time when, and the age at the date when the variety was first introduced. As I said, a lot of these varieties are from the 1960s to 2000, so we have a lot of spots in this part of the graph, but we do go back to about 1930 in these analyses. And these are some of the components we analysed, and this is in whole grain. So total phenolic acids, free phenolic acids, conjugated and bound phenolic acids, total tocols, total sterols, alcohol saucinols, phenolic lipids, and total folates. I think you'll agree there's really no trends there in terms of increases or decreases. If anything, there may be slight increases in some of these components in the more modern wheats, but because we have very small numbers of samples here, it's actually difficult to include that. But what you do see is a very high diversity in these modern lines. Look at something in the same sample set, another type of component. This is dietary fibre. As I said earlier, we're very interested in dietary fibre because wheat is an important source of dietary fibre in the diet, particularly in bread. So we determined total dietary fibre, and you see there's almost no change over this period in total dietary fibre. We looked at lignin, one of the fibre components, fructans, small fermentable fructooligosaccharides which form part of the fibre component, and another component, cellulose and beta-glucan. So no relationship here between year registration and dietary fibre content. So there's no evidence from our studies that there's been any decrease in these important components during breeding. But there is a drawback to this type of analysis, and that's the grain composition is strongly affected by the environment. In order to look at this as part of the health grain project, we actually looked at just 26 lines grown in 6 environments. Then we partitioned the variation between 3 components: the variety, the genotype, the environment, and the interaction between the variety and the environment. And you can see here from these pie charts, in some cases, the environment was the major determinant of the variation that we saw in composition, particularly phenolic acids. Other components like alcohol saucinols, tocols, strong genetic component. And in the case of dietary fibre, the genetics determines about 60 to 70% of the variation. But it does mean that you do need to look at material from multiple field trials, and health grain was carried out on one site for 1 year. But another concern about this type of diversity comparison is that the impact environment may be greater with unadapted genotypes, because when plants are grown under stress, they tend to accumulate secondary products, including some of these phenolic components. So we think it's important to really carry out comparisons with varieties which are adapted. So to do this, we went back and we decided we'd look at UK adapted genotypes only. The way we did this, we selected 39 varieties released between 1790 and 2001, I think that is. They were all bred and are adapted to the UK. And we selected them because they've all been grown in the UK for quite a while or over a large area. And they included some of the varieties we had on Broadbalk, our classical experiment. We grew them on field trials for 3 years, and we grew 3 randomised replicate plots, analysed them separately, and we milled them all to produce white flour. Now, what I've shown you so far has been whole grain. We're more interested in white flour from our other research projects because white flour is mainly consumed in the UK as white bread, and that's the major form of bread that we eat. So we milled to produce white flour. We looked at dietary fibre, particularly the 2 major components of rabenoside and beta-glucan. And to get a wider view of diversity, we looked at polar metabolites. We selected went back to 1790, and they went back up to 2012. We divided them into 3 types. From 1790 to 1916, we said these were just sort of land races, really, or selected from land races by empirical selection. So they've not been scientifically bred to any great extent. From 1935 to 1972, we had early plant breeding, and from then onwards we have modern breeding, and the introduction of dwarfing genes. You can see the dwarfing genes here coming in. 1971, mouse huntsman, there's no dwarfing genes. These three have the semi-dwarf genes, and you see they're a lot shorter. So we decided to look at the data comparing this red group here with the green group and with the blue group. Before doing this, we decided to look at genetic diversity, just to see if the diversity was greater or less. And we used high density SNP arrays. The wheat community tends to use the same 35,000 array, a commercial array, which is designed for plant breeders and for these sort of genetic studies. We do have arrays with 800,000 markers, but these are more expensive to use. In most cases, we use the 35,000. You can see the spots here, and what we do see is a lot more diversity here amongst the older varieties than the modern varieties. The modern varieties tend to be clustered on this left-hand side here of this is a PCA plot, and older varieties much scattered over a much wider area. This one of the older ones, 1838, right down here. We have one outlier in the modern varieties, Apollo 1984. This actually has DNA from rye because it's been crossed with triticale. But what we're seeing here is the effect of the rye DNA in the Apollo, in the wheat crop. But we do see lower diversity, but what we don't see is a great effect on composition. Height goes down, what we expect. This is the semi-dwarfing genes particularly coming in here, but height tended to decrease anyway. These early varieties may be well over a metre tall, and they were actually used partly for straw, for thatching cottages, and also for livestock. Height's gone down, and actually protein goes down. People often suggest that, with modern wheats with high nitrogen fertilizer, we increase the protein content. We don't, because the protein is diluted with starch. So grown under the same conditions, the protein content decreases, and we often have to increase the protein content by adding fertilizer for making bread-making wheat, for growing bread-making wheat. And then, of course, these changes in iron and zinc, particularly here in the semi-dwarf period, but also slow changes here, which are probably related to yield dilution. The dietary fibre components. Dietary fibre in wheat is in white flour, about 5% of white flour, and half of that is rabinoxylan. About another quarter of it is beta-glucan, and we can see these two components. So there's certainly no decreases, and there's probably increases with modern breeding in dietary fibre in white flour. The early varieties shown in red, the empirical varieties, then the early breeding in green, and then the modern varieties. And we consistently see increases in dietary fibre content in modern wheats compared to old wheats and in white flour. Metabolite analysis. This is a simple fingerprinting system we use quite routinely. It's simple, it's quite expensive because we have to buy NMR time. But we take flour, we extract it with deuterated methanol water, we put it in a 600 megahertz NMR, determine the spectrum, and we treat that as a fingerprint. We can either look at these peaks, which are chemical shifts, as fingerprints, or we can also deconvolute it to identify components. And we then tend to use multivariate analysis to compare composition. We get information, particularly on carbohydrates, particularly on sugars, oligosaccharides, also on amino acids, organic acids, and some other components such as choline and betaine, which can be important for nutrition and health. Just to give you some examples, amino acids tended to decrease. So these are 10 individual components summed on the left-hand side. On the right-hand side here, I've just shown asparagine. I also know, I'm sure you've noticed, we had 3 years, so we have year-to-year variation. We're interested in asparagine because it's the precursor of acrylamide. And acrylamides are neurotoxin, possible carcinogen, and there's a lot of concern about production of acrylamide during baking systems. So in terms of health benefits or avoiding sort of toxicity, these modern wheats tend to have lower asparagine than some of the older wheats. But as you can see here from the 3 years, there's probably bigger differences between the years than there are between the different ages of the cultivars. And this comes back to what I said earlier about heritability and being strong environmental effects. Total sugars tend to be higher in the modern wheats, and this may be related to the fact we have bigger pools of sugars because it's required for starch synthesis. And then choline and betaine. choline and B10 are what we call methyl donors. It means that they donate methyl groups in the homocysteine cycle, which is a cycle which actually reduces the homocysteine level, and it has benefits in terms of cardiovascular health. There's actually a health claim for betaine for cardiovascular health, although the amount you need to eat is very high compared to the amount in wheat. But it is actually used as a feed supplement for livestock. And we can use a PCA analysis to do overall comparisons of metabolites. You can see here we do get partial separation of the three groups, but in fact, you know, the three groups overlap very largely. There's a trend of early groups on the right hand side here, and that's due to these amino acids and the modern varieties here, which is due mainly to these, mainly to some of these sugars. So no major trends, and certainly nothing which we see as a decrease in any quality component. Land races are interesting because they give us a snapshot. They give us an idea of what we were growing and consuming 100 years ago. And we worked with a series of land races which were collected in the 1920s and 30s. Someone called A.E. Watkins, he asked people at British embassies to go out and buy some wheat and send it to Cambridge. There were several thousand lines, about 800 lines have recently been sequenced, completely sequenced in a project with a Chinese lab, so we have sequences of all these available now. But also we have a core collection of 128 lines, which captures most of the variation in the collection. And we've done some screening of the Watkins collection to see if we have wider range of variation than we have in modern wheats. This is just one example. This is rabinoxylan fibre in wholemeal flour, and these are 128 lines, and this is showing our control variety for high fibre, which is a Chinese variety called UMI34. UMI34 is a modern Chinese bread wheat, has the highest rabinoxylan content that we've found in any types of wheat, and you see it's very close to the highest levels in the Watkins land race. So we think there's actually very limited increase in diversity in these land races compared to some of our modern wheats. But we are working a lot now with the Watkins collection. The fact we have the sequences means we can use it to identify candidate genes, and we're working with it in terms of number of traits. Then finally, just a few words about ancient wheats. I mentioned earlier 3 types of ancient wheat: einkorn, emmer, spelt. And we did grow some ancient wheats as part of health grain. We had these 148, 150 bread wheats, and we have 5 spelts, 10 durums, and 5 emmers and 5 einkorns. So we do have some data from health grain. And this is PCA analysis of phytochemicals and dietary fibre components. When we do a fibre analysis and we put all the data into PCA analysis. You see that here, although we have some of these other cereals, oats and barley as outliers, spelt, shown in black here, falls very much over bread wheat, and einkorn and emmer also overlap with bread wheat. And we also done NMR analysis of the health grain samples, and again we can see overlaps, but also some degree of separation. In this case, the einkorn and emmer are showing a degree of separation, but they're still overlapping with bread wheat, the green spots here. So this type of analysis turns to really enable us to try and define the limits and the overlaps between species. Of course, as I said before, the health grain material is not perfect because it's single samples grown on a single site for 1 year. So we have a genotype effect and we have environmental effects here, and we have varieties grown which are outside their area of adaptation. So we've done a more sort of detailed study recently. We've done this as part of the sort of Well on Wheat project. The Well on Wheat project launched by Fred Browns at University of Maastricht, and he's a nutritionist, and he wanted to establish a knowledge base for comparing the effects of ancient and modern wheats in terms of composition, then processing and bread composition, and then effects on consumer well-being. And it's actually set up to actually do 3 feeding trials on patients on breads made from emmer, einkorn and spelt. The way we did it for the feeding trials is we actually grew 5 bread, or we actually purchased 5 bread wheats, 5 spelts and 3 emmers. and then we bulk the grain for a milling. And we bulk the grain for milling and baking because we wanted to ensure that if there was any genetic variability, we sort of evened it out. But we had to use commercial samples because we used over 100 kilos of each type of wheat. We also wanted to know whether the material that we're using was, how diverse it was, or whether compositions overlapped. So as well as using this material for human feeding trials, we also set up a G times E experiment. So we took the 13 types of wheat, we grew them on 2 sites in the UK and in the Netherlands for 2 years, 3 replicate plots, with local agronomy, but with 2 nitrogen levels. We used 2 nitrogen levels, 100 kilograms or 200 kilograms, because the 100 kilograms will really reflect what you probably use for spelt or emmer or einkorn. And the 200 is very close to what we use in the UK for our commercial wheats. We thought it was important to go out under both conditions. And one reason they use the low nitrogen for the older types of wheat is they grow very tall. These are some of our field plots here at Rothamsted. You can see some of these lines are very tall. They're well over a metre. And you can see the massive diversity we have here. It's actually very difficult material to work with and to harvest because it tends to fall over, particularly in small plots. So we took the field trials from the 2 years, 2 sites, so 4 environments, and then 2 nitrogens. And we carried out a range of analyses. Totophenolics. Here we're looking at the bed wheats, emmers and spelts using box plots here. The left-hand side here is the UK. This is the Netherlands. So very, very little different in totophenolics. When we look at effective nitrogen, when we bulk the site data, just look at nitrogen again, very little different than nitrogen on phenolics. There is a site effect on protein, but there's also a very big nitrogen effect on protein, which is what we'd expect. So higher levels at the 200, but also protein tends to be higher in the spelt and the emmer than it does in the bed wheat, and that's probably because the yields are lower and we get less dilution with starch. And then iron and zinc, these again tend to be higher in the spelt, in the emmer, but again this may be a yield effect because we get lower yields from the emmer and spelt. So we've only had this data a week or so, but we're going to now express the data on a per grain basis, and on a per grain basis I suspect it won't be very different. So these are sort of differences we'd expect, which are really based on yield dilution, I think, of iron, zinc and protein. We've also done a metabolite analysis, and emmer, spelt and red wheat, and this is the 4 metabolite data set, which I'm not going to show you, I'm not going to talk about. But I'm going to show you PCA analysis. The PCA analysis is taking a metabolite data set, and there's about 30 components. And these include sugars, so monosaccharides, disaccharides, oligosaccharides, also amino acids, 10 different amino acids, organic acids, and choline and betaine, so over 30 components. If we look at PCA analysis of the whole data set, we look at PC1, 35% of the variation, plot that against PC2, 13%, so there's about 48% of the total variation here in the sample set. We can see that we get a very good separation based on year. The 2018 samples, these are from the UK and from Flevoland, are quite nicely separated from the 2019 samples. We can also look at the same plot, but we can, in this case, colour it according to type of wheat. And we can see here that we get an overlap very much in the middle here, but some separation of the emmer in this part of the PCA plot, some separation of the spelt and some separation. So partial separation of the 3 types of wheat, but in fact there's a very big overlap in the middle here. And these samples I couldn't tell you based on composition what type of wheat they are. We can get better separation if we use a supervised type of multivariate analysis. PCA, use the whole data set. OPLS DA, we actually asked the statistical package to just look at variation associated with the difference between the cereals. So we say, let's see the difference associated between emmer, spelt and red wheat. When we do this, we use less of the variation, 17% PC1, 9% PC2, so 26% of the variation. But by doing this, we can separate emmer, spelt and red wheat based on this small amount of variation. And here we have a loaded plot, and we can see what the separation is based on. So the answer here is, I think, that these species, these types of wheat overlap in composition, but they do also have some sort of differences, and we can actually pull them out based on the differences if we try. We can also use this sort of analysis to give us more quantitative data. Here I haven't looked at the difference between species, I've looked at the differences between nitrogens. And we can do a scores plot where we look at samples, look at components which are, say in this case, high nitrogen samples, reduced in high nitrogen samples, or high in UK samples, reduced in UK samples. So we can get a lot of data from multivariate analysis on exactly which components are affected by the environment, or which components differ between the different types of wheat. Okay, I'm not sure how I'm going on time, but I think I'm probably within time because we started a little bit late. But I'd like to conclude to leave some time for discussions. So what we know is that wheat is immensely diverse. If we look at gene banks, some 200 gene banks globally, we have over 850,000 accessions, and that's accessions of triticum. So the genus Triticum, and we know we have something like 50,000 different genotypes which are grown commercially. So massively diverse, and the diversity is associated with adaptation as it's spread across the world in the last 10,000 years. So it's spread across the world and become adapted to different environments. Things like changes in flowering time, changes in resistance to frost and other weather conditions. With the exception of minerals, we can't find any real evidence that older types of red wheat differ from sort of modern types of wheat. Okay, that's a mistake. There should be modern wheats. And similarly, we think that ancient wheats have very, very similar compositions and overlapping ranges. When we find differences, these tend to be small. And in terms of effects on diet and health and nutrition, it's very difficult to conclude that they're significant. There's a whole lot there's a lot more work on these sorts of comparisons. We're particularly interested in protein composition. I haven't talked about protein, but as part of the Well on Wheat project, we do have a lot of proteomic analyses being carried out at Wageningen at the Agricultural University. So we will have proteomic data as well on this comparison from Well on Wheat. But you do read a lot of studies which show differences between different types of wheat and different types of food produced by them. I think we have to bear in mind a number of things when we do comparisons. The composition of wheat-based foods is determined partly by the genotype, but we've shown that the genotype only controls parts of the composition of the grain. Composition is also determined by the environment, the climate, the interaction of the climate with the genotype, and also the soil type and composition, and particularly the mineral content and composition. I haven't mentioned selenium, but selenium content is determined really by soil content, but contents of other minerals will also affect the composition of the grain and, of course, the nitrogen fertilization. So agronomy has a big effect, as we know. These interact, but also composition of food is affected by the way that the food is prepared. We never eat wheat as a raw grain. It's always processed, and normally to process it, we mill it, and then we make some type of product, normally fermented products such as different types of bread. For milling, there's a lot of variation in the way the grain is milled. What we call extraction rate is the amount of white flour we recover. The starchy endosperm, the white flour fraction of the grain, is about 80% of the total, and millers will normally get 78% or 80% of the grain as white flour. But if they get higher contents, they get more of the bran, and the composition changes. The composition changes with lower content. So the way it's milled, the particle size will affect the analyses, and particularly bread making. Nowadays, modern wheat is mainly made with yeast fermentation, but there's various types of system. In the UK, we use a very rapid system called the Chorleywood bread making process. Other countries use more traditional long fermentation systems. And of course, sourdough systems are becoming much more fashionable nowadays. And sourdoughs are fermented with a mixture of yeast and lactic acid bacilli. And the types of fermentation have a massive effect on the composition. So really, I think if you're comparing different types of wheat, you have to look not only at the wheat itself, the genotype, you have to look at the way it's grown, the way it's processed, and then you have to look really at what we eat. And so very often you find that when you look at the literature, comparisons are not equivalent in these different, in terms of these different factors. Either the genotypes are grown differently, or the milling is carried out differently, or they're comparing different types of products, such as a traditional loaf with a sourdough loaf. I'd like to stop there, just to thank a few people. My colleagues at Rothamsted, Alison Lovegrove, I work very closely with. She co-leads the group. Work carried out by Jack Dunn and Till Pelney on the samples, and Jane Ward leads the metabolomics group, who carries out all metabolomic analysis and helps with the data sets. Work at Maastricht was initiated by Fred Browns. We also collaborate with other people at Well on Wheat. We work very closely with our colleagues at the John Ennis Centre, Simon Griffiths and Lucy Wingan, who are wheat geneticists. And so some of the data I've been talking about has been based on studies carried out with them. Okay, so I think I'd like to stop there and leave some time for questions, hopefully. Thank you.
Thank you very much, Peter. I think for the questions within the auditorium, Christian can maybe go around with the microphone. Yes, for people who have questions online, they can type their questions in the chat, and then we can, then Peter can answer them directly that way. Yeah, I would like to first ask a question myself. First of all, thank you very much for this very interesting talk. One of the things I was really surprised by was what you were talking about with the protein content going down over time. That I thought was really surprising, that as more starch was being added to the grain. Yeah, do you have any idea yet? Because you said a little bit about that you will be starting with more protein analyses, but do you have any idea of the protein composition already in this process of going down? For instance, with something like with lysine, for instance, because it's often seen as being limiting in cereals, and especially for historical questions, that would be something of great interest.
Yeah, I mean, lysine is not generally an issue for plant breeders nowadays, because of course in terms of feed grain, we supplement feed grain with soybean or with oil seeds. And in terms of human nutrition, lysine is very rarely limiting, you know, when you wheat as a diet. What happens, I mean, the lysine content would tend to decrease as the protein content increases, because the increase in protein content is mainly due to gluten proteins, and they're actually very low in lysine. Mm-hmm. So if anything, the lower protein wheat would tend to be higher in lysine. Because it's bred for dose strength, it would tend to have a different balance of proteins. The modern wheats tend to have more glutenin and less gliadin. So they have a different balance because of the emphasis on dose strength. But the total amount, people are always surprised. But see, if you think about it, wheat is 80% starch, probably 10 or 12% protein. So inevitably increases in starch will dilute everything else, particularly protein, as the major component. Although we add high fertilizer, we add it partly to increase our yields, but also in the UK we have to add extra fertilizer to get back to the protein level that we need, because the protein level is now too low when grown under old-fashioned conditions. But we know quite a bit about protein composition. There's quite a lot published on it, and a lot of my own work in the past was on proteins.
Okay, thank you. Christian, your microphone is still off. On the computer, the sound is off.
Yeah, can you hear me now?
Yeah. Yes, now we can.
Yes.
Yeah.
Okay, so I go through the audience. Amaury, yes.
Hello, can you hear me?
Yeah.
Yeah. First of all, thank you very much for the presentation. That was a lot of information, and I'm really thankful for that. I would have a question on the trial that you talked about around the middle with the 39 varieties.
Yeah. Okay. Yeah.
Yes, this one. And here you show that generally you have a decline in protein, iron, zinc, I think some amino acids too, with the year of release. And I don't recall if you talked about the relation to yield. So could this decrease be linked to a dilution effect, or can you have some information about that?
I think a lot of the decreases will be yield dilution. I don't think there's very much we see which would be more. The only thing we see which is not solely yield dilution is minerals, because we know that iron and zinc would decrease in the semi-dwarf wheats, even if the yield is not increased. If we grow them under very low yielding conditions, they still decrease. But I think most of the other components are yield dilution.
Okay.
I think what we find is unless you select for something, it doesn't really change. You know, you select for high starch, it doesn't mean that other components will change apart from being diluted.
Okay. And would you have any hypothesis about the decline in zinc and iron? I've read for myself an hypothesis that was about selecting for whiter flour that indirectly selected for a lower rate of ashes in the grains, and that indirectly this may have declined the nutrient content.
No, I mean the nutrient content actually declines in whole grain, and we think it's because the semi-dwarf wheats are probably less efficient at removing minerals from the soil. And the semi-dwarf wheats have a short stem because they have a high harvest index, and although it's never been tested, it may be their root systems are smaller as well.
Okay, thank you very much.
They may just be less efficient at removing iron and zinc, or they may be less efficient at transferring it to the grain, but probably taking it up. But it's a good question, thank you.
Yes, you have a question? Ah, okay. Then Christoph, Orlando University. No.
Hi, yeah, so I guess just to make sure that I understand the overall conclusions here, you kind of did find that there are some important declines in the nutritional quality of wheat kind of since the Green Revolution.
Only minerals, iron and zinc.
And protein, right?
Protein, yes, protein goes down, but protein is very much controlled by nitrogen availability. I mean, I don't think the genetic potential for making protein is much lower. I think it's just because it's controlled so mainly by nutrition.
Right.
But
so
you're right. I mean, protein does go down, and that's almost certainly dilution because we have higher yields. But yeah, protein would increase if you increase the nitrogen availability. And the problem is that if you take off 10 tons per hectare, you need 300 kilograms of nitrogen to get your protein level at the level for bread making. So you need a massive amount of nitrogen fertilizer to get 13% protein in 10 tons. If you work it out, it's about 300 kilograms per hectare, much more than we normally use. And so people often complain, you know, we have these high yielding varieties, the protein's too low. But of course it is, because you know you can only take nitrogen off when you put it on, and we just can't add enough nitrogen because our yields are so high.
I see. Interesting. Okay.
But if you grow them under lower yielding conditions, the protein content will be higher. But then there's but that's not the point really. They're good. They're grown for high yielding conditions.
Right. Cool. Thank you.
Thank you.
Next question. Oh. Jorge. Jorge. So Jorge is from ULB.
Hi.
Hello, thank you, Peter. I have a question about this tendency. You think the tendency is going to change since, and it is linked with the fact that in the last decades we were focusing the production of crops, but now we are more like the breeding is more focused now on the quality of the grains, for example. You think his tendency is going to change of proteins and some nutritional values?
I think it would change, but it would change but it won't necessarily change in all countries. I mean, I mentioned earlier we have 750. You know, we produce 750 million tons of wheat a year. Well, in some countries, such as Central Asia, that's half the calories.
Mm-hmm.
I think in countries like the UK and to Western Europe, there will be more of an interest in quality. In a lot of other countries, it's still the main source of calories. And of course, places like India, where they eat a lot of pulses, a lot of vegetables. Again, there's no real problem with just using wheat as a source of calories. So I think it would depend on the culture and the economic status of the countries. In Western Europe, yes.
I was more in Europe.
And I mean, our interest, our main research interest is trying to increase the intake of fibre, dietary fibre in the diet. And because we get 20% of that from bread, we think that's a good vehicle for increasing fibre intake.
Okay, thank you.
That's a good question. Thank you.
So we go back to, no, not Amaury, Paul.
Alan. Peter, this is a question from an economist, so I'm afraid it will be a little bit naive, I'm sure. But I was struck by the number of times you made a link between nitrogen and protein content, and the fact that protein is as a proportion of the grain is falling. Within the European Union, we have a policy to try to reduce the use of nitrogen by 20% by 2030. In your view, what would be the implications of that for the dietary value, if you like, of wheat if we actually proceed along that path?
Not so much the dietary value, because although we get 10% of our protein from wheat in the UK, it's not the major protein source. We have a lot of other protein sources. The main impact is on the processing quality, and being able to maintain the processing quality at low nitrogen. Because protein quality because the protein content and quality is the major determinant of things like being able to make decent bread. So that's the main impact. We need to reduce our reliance on high protein wheat for bread making. So be able to improve the quality at low protein content, or reduce our yields. We just accept sort of much lower yields. We accept that 8 tons or 6 tons is the most that we're going to get. But if we want 10 tons per hectare, we do have to put a lot of nitrogen on for bread making wheat.
So thank you very much for your very nice presentation. I have a question related to the Charlie with the bread making method that you mentioned at the end of your talk. I've not, I don't know exactly if it is such a fast method because it uses a chemical or biological yeast. Could you give more information about this?
It's a very fast method because it does the mixing very quickly by alternating high and low pressure. Okay. I don't know about it. It does use yeast.
Okay.
I mean, it uses yeast, but it mixes very, very quickly with a lot of energy, and they change the headspace by making it high pressure and then low pressure. So it mixes in a matter of minutes, and then you have a very short fermentation period.
Okay.
It's very efficient, and it actually is sort of very good with the sort of wheats we grow in the UK, which are not particularly good bread making quality. So it deals well with poor quality wheats. But it's a very, very rapid process, but uses quite a lot of energy.
Yeah. Okay, thank you.
Okay, so now we have a question. Ramji, you said no? Amaury, here, no one? Okay, so we go back to Amaury from ULB.
Hello again. So first a question, a simple question I forgot to ask, but are these results from the comparison of the 39 varieties published yet?
Yeah. I mean, the probably best thing I do is I send some papers to Fritz and he can send them around. Yeah, they were published about 2 years ago.
Thank you very much. And another question is more broad and maybe complex question, but if we take the perspective of organic farming systems and breeding for these kind of systems, where we have different kinds of more like organic fertilizers and so on. What is your take about the relationship with the dietary quality and the potential for breeding towards more nutritious?
We do. I mean, we've done a small amount of work on organic samples as well, which is published. I'll send that paper through as well. I mean, my view is that organically grown wheat is so different, but it's not necessarily better. It would tend to have more nitrogenous compounds, more amino acids, which can be an advantage. In the case of asparagine, it can be a disadvantage because of processing and the formation of acrylamide. The main problem with organic wheat is to get enough protein for bread making. Because there's very little protein in the fertilizers, and the protein there is used, and so there's very little nitrogen in the fertilizers, and it's actually released very slowly. I mean, traditionally in our broad bulk experiment, we use farmyard manure, which contains a lot of nitrogen, but almost none of that is available. So it's a very slow release. So the main problem with organic wheat is from processing is that it's very low. It tends to be low protein.
Yes.
The composition does differ, but I don't think there's any evidence that it's better.
All right.
Depends what you're looking for.
Okay. Thank you very much.
Another question? Turn around. So in the chat, it was Antonella, but you asked for your question already?
Yeah, she asked the question already, I think.
Yes.
The Shewry would.
Okay. Okay. So we don't have any more questions here.
No.
Peter. So once more.
That's great, thank you.
Okay, 1 time, 2 times, 3 times. Done. Okay. So we thank you again for your.
Thank you very much. I've enjoyed talking to you. I'm sorry I can't be with you, but I will join you later.
Okay, we would have loved to see you here in Brussels, maybe another time. But thank you again for your presentation.
Okay, well, thanks everyone for listening and for the questions. Thank you.
Okay, so our next speaker is Jorge del Cueto Chocano. Jorge is working with me, trying to find back my. Yes, I found it. So Jorge got a PhD in 2016 in agronomy and engineering. He worked on almond and plant breeding. So then he did a postdoc at Agroscope in Switzerland, first in Sion and then in Geneva. And he worked on apricot resistance to pathogen. And then he was showing some interest on wheat, also resistant to pathogen. And it's okay. So for 1 year now, Jorge is doing a postdoc at the Crop Production and Biostimulation Laboratory. And he is the project manager of IRANET Action on Sustainable Crop Production. He will tell you about this in a short moment. So his publication record is a 16 paper, and he's a co-author of a science paper on almond domestication, where he mapped a gene encoding the bitterness of almond. And I think we are just synchronized, because Jorge is ready.
I don't know this frame. It's weird.
This is supposed to be the recording. But I don't know why it is.
It's not recorded.
Usually it should occupy the whole, the whole screen.
Okay.
Never mind. Okay, so my pleasure. Jorge, the floor is yours. If you have questions in the chat, you write them, and in the audience, I will go through you with the microphone. The floor is yours, Jorge.
Thank you, Christian. Can you hear me well? Or it's too close? It's okay? And online, it's fine? Okay, great. For once, it's working. Okay. Thank you, Christian, for the nice presentation. Today, in this frame of BrIAS between ULB and VUV, I'm going to talk about, I'm going to explain you a bit about the project that I'm doing here. So basically, I will talk about how the crops in Europe are overcoming the climate change, or we are helping the crops to overcome the climate change. So I was going to introduce a bit of myself. Christian did it, but I want to do just a bit. So I'm currently from Spain. I did my PhD in Sebastesic in Murcia, working mainly with plant breeding on almonds. So we're looking for the responsible gene of bitterness of the almond. Finally, we got it. This one? Yes. And then I moved. Well, during this period, I did 1 year at the University of Copenhagen in Denmark. After this, I got the first postdoc position in Aeroscope, Switzerland. It's a national center of agriculture. I work there with apricots, breeding programs, plant breeding on pathogen resistant. And so during 10 years, I was working with trees. I got tired a bit of the trees because it's quite difficult to work with trees, and I decided to move to cereals because I was quite interested in cereals. And then I did a small project on wheat in Geneva. And then last year, I got the second postdoc position here in ULB. Working with the crop production and biostimulation laboratory with Prof. Yes. Can I? Okay. Yeah. Oh, better. With Christian. And yeah. So what I'm doing here in Belgium is I'm working in a European Crop Internet project called C4 Future, fortifying and enhancing the resilience in C4 crops for current and future climate change adversities. Okay, very long title. So, but first I want to explain you why we call to this project C4 Future. First of all, why future? Of course, we all know about the climate change. This is a world map of the variation in the temperatures in the last 150 years, according to NASA. We can see an increase between 1 or 2 degrees, especially in the last 20 and 30 years. So the predictions we have already are that the climate is going to be drier and warmer. And if we focus in Europe, especially the Mediterranean area, is going to be very sensitive to this change of the climate, but also Central and North Europe. Second, why C4? Why C4 is because C4 plants. C4 plants are special plants different from the C3 plants that are the majority of the vascular plants. Basically, to be simple, these plants concentrate the CO2 in a compartment, so the photosynthesis is more efficient. And then the plant doesn't have to open the stomata so often, and is losing less water by evaporation. So these plants are more adapted to dry and warm environments. For example, this, and they are only, like I said, 3% of the vascular plants in the world, but still there are some important crops like maize and sorghum. Then casualty are the crops of our study, the 2 crops that we are studying in the project. Maize is the first cereal in the world, and also the main crop in production and surface. Sorghum is the 5th cereal in the world. Maybe it's more important in Africa and Asia, but it's getting more and more important, importance in Europe. They are mainly they used for human consumption, for animal feeding and also for biofuel industrial purposes. And okay, first I present this Q4 and future. My next question to you is, which are the two most important limiting factors for a plant growing, apart of the light? And the answer is nitrogen and water. Of course, I don't have to say, you all know about the importance of the water nowadays, and in the future it's going to be more important, a real limiting factor. And the nitrogen, the plant needs some micronutrients, but the nitrogen is the nutrients that the plant needs in more quantity. And how the plants take these two components. And of course, the answer is the roots. And during the domestication of the crops, during the crops domestication by the humans, even breeders in the last decades, we were always focused on the upper part of the plant, like the production, the height of the plant, different parameters. So, but we forgot a bit the roots, and the roots are very important because the plant is taking everything from the roots. So, in the breeding programs, we should take also in account the roots, and there are a lot of things to study yet from the genetical point of view, from the mechanism that regulate the system of the roots. Okay, so I'm going to explain you a bit how the roots interact with the nitrogen content in the soil. That, for example, here we have a plant in a low nitrogen content soil. So the plant, because it needs to eat, so it's looking for the to eat, like it's looking for the nitrogen, and is developing the roots. In a second image here, we can see soil with a high nitrogen content. The plant, of course, is bigger than the other, but the root system is less developed. So let's say that the plant is getting lazy because it has everything that it needs there. But there is a problem with the nitrogen, as well, I didn't tell you that the plant is not able to take the nitrogen from the atmosphere, but is able to take in organic way, nitrates and ammonium way. The problem with the nitrogen fertilization, and we were discussing before, is like there's a lot of contamination for this nitrogen fertilization. So the leaching of the nitrates, because they are not very stable in the soil, they can go to down the soil and contaminate aquifers and water that we will use later for us or for the irrigation. So problems of eutrophisation, and also problems because the part is coming back in form of CO2. So we have to reduce the inputs of nitrogen, and also because the European law is tending to reduce the nitrogen fertilization. So from the genetic point of view, we are looking for this ideal plant with a good production, but also with a strong root system with a less income of nitrogen or water. And these are the concepts that I'm showing you here, like resource use efficiency, nitrogen use efficiency, and water use efficiency. It means that, for example, for the nitrogen is the quantity of nitrogen that the plant is converting into biomass. And we are trying to increase this efficiency of the plant. So the plant is becoming more resistant to water and nitrogen deficiencies. So these are the goals of the project. The main goal that is not a goal of the project is beyond the project is how can we sustainable ensure harvest security under more variable climate conditions. For this, we have these goals, different goals of the project. For example, determine the variability in these crops in response to nitrogen and water efficiency, stress. to identify the genetical regions responsible for this resource use efficiency, and also parameters and characteristics that we are able to detect. And finally, we are going to do a bit of genomic prediction and selection, and finally, develop a toolbox to breed the C4 cereals. Is because I didn't tell you the project is 3 years, but it's a bit beyond the project, but we can try to find some molecular markers for the breeding programs. Okay. The structure of the project is a project of 6 countries, 6 different institutions in Europe. These are the main responsible per each institution. There are more people behind, of course. So me and Christian in ULB, then INRA, Paris and Montpellier, Alain Sarkozy and Romain Chapuis, Efren Abi, Amana, and Increa Bologna. I will explain about him later a bit. Fahim Balog in Sivas University in Turkey, Stefan Gerth in Germany, and in Aarhus University, Luke Jans and Line Carlenius. And this in yellow, we can see the 3 fields that we have for testing our crops. One is in INRA Montpellier, the other is in CREA Bologna, and the third is in Sivas University in Turkey. This is the structure of the project. It's a classical project. It's let's say nothing new from the structural point of view. For me, the most important of this project is we are talking about cereals, we are talking about water and nitrogen stress, and about the roots. So it's more the object of the study, more than the structure of the project. First work package is the one that we are doing here in Belgium. It's about phenotyping and control conditions. Then the second work package link with the one that we are doing here is phenotyping on the field. And with these 2 phenotypings that we will do during 2 years, then we will do the pre-breeding research, the genetic approaches. And then the 4th and the 5th is the dissemination and the management of the project. I'm gonna explain you more a bit in detail what I'm doing here we are doing here in the lab in ULB. So for the plant material, we received last year 2 panels of maize and sorghum of 280 inbreed lines and 180 breeding lines from INRA and in France and CREA in Italy. We are using this material for the whole project, of course, for the phenotyping and the control conditions in at early development stage, and for the field trials. As you can see here, already in the seeds, we see differences between these aligned repetitions of the different lines, and we see already differences in the color, in the size of the seeds, and the same for sorghum. So we are doing mainly 2 different tests and control conditions, as I said. One test is on roots. The other test is in the upper part of the plant. For test on roots, what we are trying to measure is we are trying to do a image and image of the plant, of the architecture of the roots. For this, we germinate the seeds. We always work in ideal conditions for the maize and sorghum growing, and always with hydroponics, means without soil. So we give the plant everything that it needs from the nutrient point of view. We work with this poach and wick system. You can see here. So it's we put here the seeds, germinated seeds with a germination paper, and we cover with these 2 panels of plastic. And after 4 days, we scan the plants, and with a special software we measure different traits of the roots. These are the like the main traits that we are observing or we are studying, but there are a lot of parameters inside these traits, but I didn't show, but we have much more. So mainly we are measuring the principal root of the plant, seminal roots and lateral roots, and also the biomass. You can see here roots of sorghum and roots of maize. They have big, the main difference that they have is that, for example, in maize, we have these principal roots and the lateral roots, and also we have some seminal roots. While in sorghum, we only have the principal root and lateral roots. It's a characteristic of each crop. Okay, this is the test on roots. For a second test on the upper part of the leaves of the plant, of course, because they are related, the root on the upper part of the plant. So we basically measure what is happening there, the stage of the leaves, the stage of the plant related with the roots. So we work again in ideal conditions, in hydroponics, and we do 3 treatments: control, nitrogen deficiency, and water deficiency, and also we do kinetics. So every week we measure different parameters like biomass of the roots and the shoots, photosynthetic activity with this device here, a fluorometer. So we measure different parameters of photosynthesis of the plant, and with this device here, the polypen, we measure also the reflectance of the leaf. So we can measure pigments of the plant, the nitrogen balance index, and other parameters. Okay, and this is the different phenotypings that we are doing under control conditions. So we work with, as I say, with roots, shoots, and control nitrogen and water deficiency with sorghum and maize. We decide because here for sorghum you have nitrogen water deficiency with the root test. We are doing maize because at the beginning we thought that 4 days it was not enough to see a difference in the roots of the plant of maize and also of sorghum. But we had a small project with some bachelor students here, and we decided to see what is happening with sorghum. So we decided also to do with sorghum roots. Okay, and this in red is because we are still in progress. So they are the tests that we are going to do in the next 2-3 months. Some results of the preliminary results, because we are still working on it. First of all, we are looking for, of course, we have 2 panels. So the first thing that we look is we look for diversity, because if everything is the same, we cannot do anything for the genetic part. So yes, we have diversity. So I try to classify the different genotypes in different patterns. Like, for example, this genotype here is like a match that we obtained from the software that we studied. So this genotype has a principal root long compared with the other. We have several seminal roots and a lot of lateral roots. You can see here all the numbers of this genotype. These ones, for example, long principal root and seminal roots, but no or few lateral roots. The same for this and the same for this. But for example, this we cannot see. We don't see any lateral or seminal roots. So at the end, we have different patterns of the roots. This in maize. The same here, but these main roots are all shorter, like between 3, 5 centimeters. The same for sorghum. We see differences in the panel of sorghum. So here we can see this genotype with long principal roots and with a lot of lateral roots. But for example, this has lateral roots, but the length of them are very short compared with the other. Okay, first, diversity. The second, we wanted to know what is happening with this nitrogen and water stress in the plants. So I'm going to show you some examples of genotypes that we observe an effect. For example, this genotype, these control conditions. So we have the root, this sorghum. We have the roots like this without almost nothing on lateral roots. But when we induce this nitrogen or water stress, the plant, the roots develop lateral roots in number and also in length. So in this genotype, we see an effect as I was explaining before. And for this, you can see the control, and with the two stresses, the plant is reacting to this deficiency. The same for this. But when we go to the main panel, here again we are just talking about roots and about the sorghum. We see, for example, in the principal root a decrease with the water and nitrogen stress. Then same for the number of lateral roots, and length of the lateral roots are more or less the same. We expected a bit different tendency, like the lateral roots they were to increase, and also the length with these stresses. We're still working on it. So this sorghum we are doing this month now. We finished today. The same test on maize. So maybe we'll see a different tendency. And the same for the root biomass. We didn't observe really a significant increase of the biomass. Then we go to the upper part, still working, talking about sorghum. What is happening in the upper part? This one parameter, NDVI, Normalized Difference Vegetation Index, is basically talking about the state, how healthy is the plant. So if this is 1, is very healthy. If it's 0, is dead. And here we see differences between the control and the nitrogen deficiency on the sorghum and the sorghum panel. Again, the root biomass is decreasing, and the shoot biomass, shoot is all the part this upper and above the roots in the plant. And also talking from the photosynthetic point of view, we measure the efficiency of the photosystem too. And we didn't observe big differences between both the control with the stress. And it's something related with the last talk of the day of Christopher, that you remember this pathogen that we didn't observe like a differences in the photosynthesis. So here the same. Photosynthesis is similar, even that the plant is under a nitrogen stress. Okay, some correlations. Of course, we have much more parameters than this, but I try to select the most interesting for this talk. So we see some. It's a bit difficult too, but here you have the legend. Like we are taking all the parameters that I was talking before and putting together. We see some correlations, for example, between the principal root and the number of lateral roots and the length of lateral roots, what is quite logical. Also, we have some also correlations between the root biomass in control conditions with the root biomass in nitrogen or water conditions. What is also logical, because I was saying before that we didn't see really differences between the stresses in the biomass point of view. So Yet we don't have very good correlations yet, but we're still in progress, so we expect to have more interesting things. So this is the part that we are doing here in the lab. Now I'm going to explain a bit what my other colleagues are doing, because they cannot come, so I'm going to explain everything. Basically, we continue with the phenotyping under control conditions, but this time with soil. So we already select some genotypes, and we are going to send more seeds to our colleagues in Germany. Basically, they are going to do a 3D system architecture in soil through computed tomography. So they have this room with that they can control temperature, humidity, and light, and CO2, I think, as well. So they are going to we send like 20 different genotypes. So they will put this the genotype in pots with soil under control, under nitrogen and water deficiency, and they will do this root cementation and 3D image of the plant. So we'll have like a complement to our 2D architecture. Also here we will send to the fields, I think to 1 or 2 fields, these tubes of plexiglass, I think it's called. So also they can do this 3D image on the fields with this root image system. Okay, this what I explained is about the control conditions. Now we go to the field. So we have mainly 3 fields. One in INRA Montpellier, as I explained. Second in Italy. Third in Turkey. The one in Turkey, as you can see, they had some problems this year, well, the last year, because during COVID they couldn't go to the field. There was a problem with irrigation, and they lost all the crop. So we don't have results from them yet. But we sent some seeds, so for this year and the next, we expect to have a good crop, good evaluations. In CREA, we our colleague evaluate maize and sorghum and the hybrid, and only under hybrid stress, measure different traits of the yield and photosynthetic traits. The problem is the responsible of CREA had got a position in India, and then so we are going to do the second evaluation in India with the same panel. According to him, it's okay. We can measure because it's never the same environment. So we'll see what is giving is not European anymore, the project. And then we have the results from India, so from INRA, working on maize and nitrogen and water efficiency. So they are measuring different parameters of the at the experimental level, like sowing date, emergence date, plot level, like the vigor of the plant, plant numbers, harvesting date, the kernel weight, so different parameters of the plant and the kernel. And at plant level, you can see here air leaf rank, final height of the plant, and they are using these drones to measure the leaf area index and the final plant height. Also, they are measuring doing this image of the years to evaluate for example, the grain number per year, and with this NIRS machine measuring the protein content of each plant. And of course, it's also important to do a soil analysis to see nitrogen content in the soil, because if we are talking about nitrogen stress, but then we have nitrogen in the soil, so we are not really in this nitrogen stress. So yeah. And then with all of this phenotyping, we go to the genetic approaches. We'll start this year, and we have all the first results of the phenotyping. Basically, 3 strategies. The first strategy is the association study, because we have these 2 panels of maize and sorghum. So with this lab phenotyping and the control conditions and the field phenotyping, the good news is that we have already the genotyping of the panels. So we have a lot of SNPs, difference in the nucleotides among the different accessions. So with this, we will use a mixed linear model. Model that is going to be multi-traits, because we'll measure a lot of parameters, multi-site as well, and well, we expect this to have these QTLs of genetical regions responsible or link to these traits of efficiency that I was explaining before, or let's say resistant to the plant to water and nitrogen efficiency. Also, with these regions, we have inside these regions some candidate genes. The second strategy is a transcriptomic study. So with these contrasted accessions that I was showing before, we'll validate this the candidate genes that we think are interesting through qRT-PCR to find these differential expressed genes, link again to the efficiency of the plant. We think also maybe to do a RNA-seq in complement to these approaches. And the last, the genomic prediction, INRA is going to be in charge of this. So genomic prediction basically is that we can predict the phenotype that genotype is going to be in the future. So I mean, with the genotype, we can predict the phenotype of the plant, how it's going to be in some traits. This is interesting, for example, for the plant breeding programs, because in a breeding program you do a lot of crossments. You have hybrids, and you can select easily and fast which hybrids are going to be more interesting for you. And also with this climate change that we were discussing, which, for example, if a genotype is going to be good in this environment or other without testing there. So we can predict what is going to happen with these genotypes. So conclusions and what we are going to do this year. So main conclusions in general is we found diversity in these 2 panels of sorghum and maize. We saw some potential genotypes that can be interesting for our genetic study. We have almost ready the first phenotyping for doing this GWAS association study. Of course, next year we'll have more data, more robust data, because 2 years better than 1 to have these results, but we can start already. And we can say that we have already some indicators for this efficiency of the plant against water and nitrogen stress. For this year, we'll finish the phenotyping, I hope. Data analysis, and we will start this association study and some transcriptomics analysis. This is our website. This is the link of the website of the project. Is Denmark, because they are in charge of the dissemination part of the project. So you can enter and see more in detail what I explained here. Well, this is our team. Not everybody, because we have more master students, more people there, but it's basically our team, crop production and biostimulation, the two Chief Christian and David Canela. And this is, well, thank you. This sorghum crop of our colleague in Turkey. It's not exactly the panel that I saw, but I found very beautiful pictures, so I put it just to show a bit the sorghum, to a bit of promotion of the sorghum. So yeah, I did. Yeah, it's good. So thank you for your attention. I put better down. Yeah.
Thank you, Jorge.
Water.
Some water?
No, no,
we don't have.
Yeah, no, that.
Yeah, but I don't know. Some question in the audience. Are there any?
It was very clear.
Jeroen? Yes. Ah, you were pointing at.
You want to go with the computer?
Ah, okay. Yes. Okay.
Yeah.
Okay.
George, thank you very much. This is again going to be a very naive question. I leave the more technical questions to those with the expertise. So I'm really trying to understand the motivation of the project, which you set out very well at the beginning, but just to make sure that I understand. So in your first 2 or 3 slides, you showed us a picture of
plant.
two plants with low nitrogen and high nitrogen. And I'm assuming that the high nitrogen is not something that's intrinsic to the soil. That's where nitrogen has been added. So we've added nitrogen, and then you indicate that you have these losses either to water, to the nitrates, or to the atmosphere through the nitrous oxide.
Yeah.
So then your point with the third diagram is that if you can try to encourage plants that have this more bushy or dense root system, that somehow we could improve the efficiency of the nitrogen use or intake. And I'm not sure I understand why that would happen. So in other words, your second plant is making, it has all the nitrogen it needs. It's growing. It's a very productive plant. It has these negative side effects. So you then create a plant that has this bushy root system. So how is that going to? I mean, the plant is already taking up as much nitrogen as it needs. So how is the more bushy root system going to, in a sense, avoid the loss of the nitrogen through the two roots? I wasn't quite sure how that's going to happen. Is that a question that you can understand, or have I made it clear?
Yeah, you mean how we are going to create or to find?
Well,
this,
just what is the mechanism that's when you have your bushy? So you explain through the phenotyping and ultimately the genetic modification, you're going to create or hope to create this more bushy root system. But how is that in itself going to address the nitrogen loss issue? I couldn't quite see that element of the story.
Yeah, we have two things here. Well, of course, I explained that the problem is the nitrogen contamination. Yeah, so contamination of the water, contamination of the atmosphere. So, and also the law is going to be more and more strict with this. So the limitation is we have to limit the nitrogen content or fertilization. There are two things. One is to create, and the other is to find in the germoplants a good. So it's what we are doing now. We have this panel of different genotypes, and we are trying to find the best genotypes. Yeah, the best genotypes that they are already with a big root system, but also a good production. So the first is to find what we have in the nature and trying to find a good genotype and try to find the best genotype for the best environment. That's why we have these different field trials. And the second is to create it. for the creation of the genotypes, we need to know the genes that are involved in the for example, this root development.
Okay,
is what we are.
Let me, if I could just follow up with just, so is it that with the ideotype, the third
Yes.
Picture, that somehow with the more bushy system, the plant is more efficient at absorbing, if that's the word, the nitrogen, and therefore you don't need to apply as much nitrogen. Is that
exactly
the sort of, okay? That's what
I
was
this
trying to. Yeah. Okay. Sorry, that was really what I wasn't. Yeah. Okay.
Yeah, because normally, because the plant is, let's say, taking all the energy to develop the roots because it's looking for the nutrients.
Yes.
But it's losing production. But our goal is to have this robust root system, but also a good production of the plants.
Good. Yeah, I understand.
This is a question of probability to take. Yeah. Okay, another question. Yes, Amaury.
Yes. Actually, I have two questions, but I will start with one.
Okay.
Yes. So you do account for genotype and environment interactions, both with the ex situ experiments because you have different conditions, and also with the in situ experiments because you have different sites.
Yes.
I don't know if you already have results from the field trials, but maybe you said it. I'm sorry, but how much do you expect that? How do I say this? Yeah, how much further genotype interaction that you cannot capture just in the lab do you expect to see with the field trials? I mean, yeah. I mean, how well do you think you are able to predict this with the lab lab trials?
Yeah, that's the point. We don't have yet. We have some raw results, but they have still to work a bit in the because they got like a few months ago, but we are still working on that. So we have some results. I didn't want to show yet, but of course the idea is to why we are doing these trials on the lab conditions is because we are working with ideal conditions. So in the field, of course, you have the interaction of the environment, and we're trying to minimize this environment effect. And what the best or the ideal would be like, okay, we find this, for example, 10 or 20 interesting genotypes, and that they will find the same in the field. So this is that would be the ideal, just to have similar results.
Of course, but I didn't really phrase my question well, but yeah, the main question was, do you see that it fits quite well for now, or?
Okay. Yeah. I cannot tell you. I cannot tell you. I mean, what they is they gave us is like a general result, but then I have to go like more in deep, like each per each genotype. Yeah.
Okay. I'll have to ask you next year.
Yes. Next BrIAS, next year I will answer the question. Yeah, but of course, that's the point, the main point of the the phenotype.
I still have another question, but if someone else has a question, I can pass the mic.
No, you have already the mic.
Okay, so just now this is going a bit beyond really your project, but I'm working on on wheat, but on variety mixtures and on-field diversity, if you will. And one of the things we don't really know is there really potential for diversity in root system to translate into positive interactions? So for example, is there sufficient diversity within one species to have sufficiently different root systems for maybe lessening the competition or such type of effects. And it's a question that I often have to myself. I am not looking at the root systems, but really, can we expect that it can happen? Or, and as you are working on the diversity of root systems, maybe you can have some idea on this.
Casually, like 1 month ago, we submit a project about intercropping. This is a huge European project, but we are doing the part of how different roots are going to interact. So so I think we were we wrote about the maize and the soybean. Because we think they are going to have a good not competition, like a good
complementarity.
I don't know how to say, like a positive interaction. So that being also a good point. Like not only to work with the same species, that to intercrop species. So that can be also a stimulation of the roots, I think.
Well, my question is, between two species, I think obviously you can have very high differences, depending on the species, of course. But within one species with different genotypes, you show, at least on the pictures, sometimes quite striking differences. But
But this is another project that we are working on it, so to see, how they interact with other species.
Great. Thank you very much.
That's brilliant. Alan again.
May I have a follow-up question to my previous one now that I think I understand this mechanism? So would you at this stage have any idea? So you're saying that if you could develop plants that have this bushier root system and therefore a greater probability of being able to absorb the nitrogen, you need to you would be able to use less of it, at least to apply less of it to the field. Do you have any idea of how big an effect could this be? Would you say 2% of your nitrogen or 20% of your nitrogen if you could?
Which percentage we can reduce of nitrogen?
Exactly. Yeah. I mean, is this going to be significant, or is this sort of a niche result?
For example, in the laboratory, we work with like 10% of the ideal nitrogen. So we have like it was like 1 millimolar, and we try with 10 times less. And there are some plants that they are reacting and they are working well, even with 90% less than the ideal nitrogen. And then we are also in the, I cannot remember now the figures of the field, but this can be something like this.
That's very impressive.
It's preliminary, but still can be. But this is a main problem here with the nitrogen fertilization. No, Ramsey? Wait. No,
Okay, so we're gonna close here. I thank you for your time. We thank one last time Jorge for his presentation.
Thank you.